Hi, my name's Guy Riddyhoff. I'm from Science Magazine, one of the editors there. And I'm going to be talking with Angelika Amon from the Koch Institute, MIT. And we are going to cover aspects of cell division and what chromosomes do during cell division at a basic level initially and in model organisms. And then we're going to talk a little bit about perhaps what you could consider the more critical aspect of the whole topic is, which is implications for health and disease. And then perhaps at the end we might get time to talk about women in science, which is of course an important, somewhat divergent issue, but we'll get to that and see what happens. Angelika, so tell us about chromosomes and cell division and what happens so that we can understand why it's important to know the
how
fate.
it works.
Yeah.
So the DNA, which encodes all the information to build an organism, is these threads of strings of basically information. And it's during part of the cell cycle, before you want to divide your cell, it looks like a spaghetti soup, right? There's all these strings attached, swimming around in that soup. But if you want to after you copied them and you want to divvy it up to put it into two daughter cells, you need to package them into travel-friendly units, right? And so for that to happen, you first have to compact your chromosomes, you have to condensate them, and then you have to attach them to an apparatus that segregates them. And once you've accomplished this, then you can actually go through the process of segregating them and dividing them accurately. And what we and the field in general are fascinated by is that this is an unbelievably complicated process. And cells do it with such ease, and more importantly, with such accuracy, that it leaves us baffled. And so we want to understand how on earth have we evolved to get it right virtually every single time, 10 to the 13th times, to build you or me, starting from a single cell, a fertilized egg.
So how many chromosomes does a human cell have?
So we have 46 chromosomes, 23 pairs. You each get a copy from your mom and you get a copy from your dad. And that's actually the other thing that we're interested in. In order to produce a new human being, obviously if you have two pairs of each, if you would just put two of those cells together, the next generation would have 4 copies, right? So what you need to do is you need to have a cell division that basically halves your chromosome content to make sperm and egg that then have half the amounts of chromosomes, 23. But when a sperm and an egg fuse, you'll come back to the original 46 and you can restart the division process. And this cell division that is a specialized cell division called meiosis. And that cell division is also a great interest of ours. We're really trying to understand how sort of the cell division, mitosis, that builds an organism from a single egg, how is that modified and changed so that then you can bring about this highly unique cell division that gives rise to sperm and egg. And that process too occurs with enormous accuracy. However, it's not quite as accurate as the mitotic cell division. And errors occur much more frequently. And many of the birth defects that people hear about, for example, Down syndrome, are the result of an inaccurate cell division that gives rise to a sperm and an egg. And we really are still trying to figure out what it is, why it is that this type of cell division doesn't work as well. Another fascinating question that we are deeply interested in.
So meiosis makes germ cells, sperm and ova, and they're haploid. And obviously that's critical because when they join up, they then make a diploid cell. And then that diploid cell goes through a whole series of mitoses as opposed to meiosis, where you simply copy the chromosome number each time and you don't change its actual, whether it's haploid or diploid. And you keep talking about accuracy, and the reason why accuracy is so critical because of course each chromosome is unique has its own identity and has its own set of specific genes which aren't found on other chromosomes. And so any one chromosome is absolutely critical because it will have critical genes for the development of the organism.
Absolutely. I just spoke about Down syndrome, trisomy 21. So that's a disease where a human being has, instead of 2 copies of chromosome 21, has 3 copies of chromosome 21. And that has a profound impact on the health and abilities of this organism. So that, I think, is a terrific illustration for how important it is to segregate your chromosomes accurately. If you make mistakes, bad things happen. Down syndrome is only one aspect of this importance to human health. Of course, when chromosome missegregation happens during the normal mitotic divisions, that can lead to diseases such as cancer. In fact, a key characteristic of cancer cells is that they do have the wrong chromosome composition, that they're losing and gaining bits and pieces of DNA, and that allows them to sort of escape the controls that tell the cell to divide or not to divide. And that can lead to cells growing excessively, forming a tumor, or growing in places where they are not supposed to grow. And so another deep interest of our lab is to understand what happens to cells in which the normally accurate process of chromosome segregation fails, and what happens to cells that end up with the wrong chromosome composition. That's a condition known as aneuploidy. And as I said, it has significant impact on human health. And more recently, something that we're very excited about is there's also the possibility that it could be a cause of aging. There's more and more evidence in the literature to suggest that the degree of aneuploidy, the degree of the number of cells in a body that has the wrong chromosome number increases with age. And some researchers suggest that could actually be a cause of aging, something that needs to be sorted out, but it's certainly another very interesting and exciting possibility that merits further investigation.
So when you talk about aneuploidy, you're talking specifically about a cell that is either gained or lost a whole chromosome, not part, not a fraction, not recombination, an entire loss or gain of a chromosome. So how does that happen? And can't the cell protect itself against that, or kill itself if it discovers that it's lost an entire chromosome?
You would think so. But so far, we are lacking evidence that is the case. So it seems like what cells have chosen to do is they have chosen to optimize the process of dividing chromosomes to such a high fidelity that perhaps having a mechanism that culls or eliminates cells with the wrong chromosome number afterwards is perhaps not that necessary. So the processes that are making sure that chromosomes are segregated accurately are called surveillance mechanisms or checkpoints. And it's a really nifty process where cells can monitor whether or not chromosomes have attached to the apparatus that segregates them correctly or not. And they will only initiate the chromosome segregation process until they are absolutely sure that every last chromosome, every last 46 chromosomes have attached to this apparatus called the spindle accurately. When a chromosome missegregation event happens, and it does happen, the chromosome missegregation rate is about 10 to the minus 5, we have no evidence to suggest that actually will lead to a response where these cells are either killed or basically stopped in their tracks, that they won't divide anymore. They are, however, a lot less fit. They have fitness defects. They have a lot of stresses that we and others have characterized over the years. And so over time, if they were to continue to proliferate, they would lose out compared to the normal cells that have the correct chromosome number. But once you made a mistake, you're pretty much stuck.
So I guess the consequences are different depending on what type of cell division it is. In meiosis, if you generate an aneuploid germ cell, a sperm or an ova, the consequences of that, if it survives and is used, it's that particular germ cell that forms the organism, then the consequences are drastic for the organism, because every single cell in that organism will have an unbalanced chromosome number. Whereas in mitosis, it's slightly different because that's part of our soma. So you could be like a chimera, effectively. I suppose effective. That's what, in a way, cancer is.
Mosaicism.
It's mosaicism. Yes, is that you have a small population, potentially growing, of cells in the body which have the wrong number. And that's this interesting point that you raise about, if you have an aneuploid cell in a population of wild type cells, why isn't it kind of squelched and got rid of? But because obviously it isn't in some cases.
It isn't. But if that aneuploid cell was a stem cell, for example, that whose job is it to divide and to divide and divide. I mean, over time, the contribution of progeny that aneuploid stem cell would contribute to the overall soma would be less than that of a euploid one, because these cells are less fit, they proliferate less effectively, they are stressed, they have problems. However, once you're a terminally differentiated cell and you're sort of an aneuploid cell in a tissue, you're just sitting there, you really don't have to do all that much other than sort of doing some basic housekeeping functions. And so I think in a terminally differentiated tissue, aneuploidy is to a certain degree tolerable. Of course, only to a certain level. There are diseases, there's a rare disease that causes an increased frequency in chromosome missegregation in humans, and the people affected with this disease obviously have quite a lot of problems. If the entire organism is aneuploid, because the germ cell that has created that human was aneuploid, then of course the consequences are dire. I mean, virtually all chromosomal aneuploidies are lethal. They will not a fetus with that wrong chromosome number will not develop to term and will be spontaneously aborted during pregnancy. Depending on the degree of aneuploidy early in pregnancy, often women don't even know they are pregnant. Sometimes, as in the case of trisomy 21, Down syndrome, these children will survive to birth, but that is really a very rare and basically unique to Down syndrome chromosome, trisomy 21.
So if your aneuploid cell can survive, what are the consequences for the cell? Obviously, you've either lost or gained a lot of genes. Mm-hmm. So you're getting effects on transcription. Or gene expression at least. So
it appears that chromosome losses and gains directly impact gene expression. So if you gain an extra chromosome, you will make, instead of 2 units of gene expression, you will make 3 units of gene expression. Similarly, if you lose a chromosome, that will reduce the expression of the gene, because you now have only 1 copy instead of 2. And so this, of course, really creates havoc in the cells. I mean, it really fundamentally changes the composition of that cell. And that is quite problematic. And bad things happen. And of course, you have to, as you mentioned, you have to distinguish a little bit about chromosome gains and chromosome losses. Chromosome gains, if you have extra stuff, that's a little bit, it's easier to deal with than you're actually losing stuff that you have to compensate for. So generally speaking, when you lose a chromosome, the consequences for the organism, for the tissue, for the cell are much more dire, because it's some processes, it's basically very hard or impossible to run on half the amount of gene dosage than is normally necessary. What happens when you gain a chromosome and you gain extra stuff, extra RNAs and extra proteins, that usually the cells have some capacity, some quality control capacity to eliminate these kinds of excesses, but over the long run, it's not 100% successful. So over the long run, these, for example, extra proteins that are being made, they start accumulating, they start misfolding, they start aggregating, and that obviously causes problems for protein homeostasis.
You mentioned cancer several times, and that aneuploidy is a prominent characteristic. Put it in context. Is it something that happens early, late? Is it driving? Is it something that turns an initial phase cancer into something much more dangerous, metastatic?
Guy, that's a really critical and important question. Sort of, is aneuploidy a driver of tumorigenesis? When is it a driver of tumorigenesis? Throughout the process, early or late? And there really, we're still beginning to understand when aneuploidy is important. The facts are that if you look at early malignant lesions, or even pre-malignant lesions, and you ask, are these types of lesions already aneuploid? Do they have the wrong chromosome number? The answer is yes. A few chromosome gains, a few chromosome losses, nothing drastic, but it has already happened. And so that, from that correlative evidence, one would be inclined to think that there is a causal relationship between disease and an aneuploid karyotype. But really, the experimental evidence is not as clear cut. When people start building mouse models that sort of simulate increased chromosome gains and increased chromosome losses, simulate or produce, thereby producing higher levels of aneuploidy, when you ask in these mouse models, for example, do you get cancer more easily? Is the disease more aggressive? And so forth. The answer is not yes. It's a mixed bag. Sometimes it helps, sometimes it doesn't, and it seems to be very dependent on which tissue you're looking at and so forth. So the relationship between a wrong karyotype, aneuploidy and cancer is not a simple linear relationship. And, the field is really trying to sort out what exactly, if any, the contribution of the aneuploid condition is to cancer. Our lab, our work would suggest that generally speaking, aneuploidy adversely affects fitness. So we've done a number of experiments that would suggest that proliferative capabilities of all types of aneuploid normal cells and aneuploid cancer cells is decreased compared to euploid cells. And so we don't think that aneuploidy per se is a pro-fitness, pro-survival stimulus that sort of drives tumorigenesis. But what we instead we believe is happening is that aneuploidy basically increases variability that it causes because you change gene dosage. Cells will now have different characteristics that under some circumstances might be advantageous, though generally they're disadvantageous. And the other thing that we've discovered is that really aneuploidy causes all sorts of other additional changes in the genome. It's what we call mutagenic. It increases genomic instability and causes all forms of alterations of your genome. And we believe that it's this ability to mutate the genome that is a driver of tumorigenesis.
So a quick question before we maybe speak for a few moments about women in science. From what you said, does that mean that addressing aneuploidy in terms of treatment, medical treatment, would be very difficult, something far in the future? Both in terms of women's reproductive issues, and also in terms of treating cancers or other diseases related to.
So with respect to fertility treatments and so forth, the single cell analysis, karyotype analysis that you can now do on very early IVF embryos has been so advanced that I would argue that in the long run it's probably much more economically feasible to just test the embryos and then pick one that has a right set of chromosomes. In terms of cancer, we are exploring the possibility of developing aneuploidy as a therapeutic target based on the fact that they have so many stresses and many types of aneuploidies share different stresses. And we are intrigued by developing it as a therapeutic target simply because it's just basically a universal characteristic of cancer. And so the industry, the pharmaceutical industry, is going more and more into more and more specialized and specialized and specialized treatments for as ever more smaller slice of cancers, personalized medicines. That is sort of where the industry is going. But I think we should also consider the possibility that to go after these more general and universal traits of the disease. I think they are equally viable targets, and they would have a much broader spectrum efficacy. And for those reasons, we are actually in the lab actively pursuing possibilities to look for compounds that selectively inhibit the proliferation of aneuploid cells. And time will tell whether this is a viable path forward, but I think it's certainly worth trying.
So, just to finish with the science for a second and talk about women in science. You, of course, are a woman in science.
I am indeed.
A successful one, otherwise you wouldn't be here.
Well,
we can argue about that.
Yes, we can argue.
So, what do you think needs to happen for more women to be successful, and for the women that are already in science to be more successful? Or is that's a tough question.
It is a tough question, but I think it is my personal experience that raising the number of women in science at a particular institution, putting them into prominent position, up-eases people, right? They see...
What's up-eases?
Up-easing means... No, not up-eases. It makes them feel good about it. Is that the right... Sorry, I used the wrong word.
Okay.
It makes people feel good about... it makes it a normality. That's, I guess, what I want to see, right? So you're not sort of the odd peacock out, the token woman who speaks at a meeting. If half the speakers are women, after 2 years, people won't blink an eye anymore, right? I mean, I grew up in a very male-dominated country in Austria, and I remember very clearly that my grandfather was of the firm opinion that women, simply because they're women, are terrible car drivers. Which is, you laughing. That's what I heard. And I was a 5-year-old child, and I sort of assumed women aren't very good drivers. I didn't question it. My grandfather kept saying it every time I drove in the car with it. Obviously, nowadays, nobody in their right mind would think that. And I think that's the same with everything. As long as you just increase the presence, underrepresented minorities of women in a particular field, in a particular job, in any particular environment, it'll become normality. And I mean, the same is true for MIT. Until 1970, there were hardly any women students. Now we have 50% of those undergraduate students are women, and it's completely normal. And so my personal opinion is that it's sort of increasing the presence of women in science at every level, especially at the level of a professor and team leader.
Do you think
very conscientiously? I'm personally a firm believer in affirmative action. It works, right?
Do you think that you think there needs to be any special catering for women? I mean, especially with regard to, fitting in, having children with career, or do you think that women and men can be considered equal on that, or should there be, creches at work, 6 months? Well, I guess there is 6 months for maternity leave.
There's a 1 year, you 1 year maternity leave for any parent. So I think in our times now, men are much more involved in the rearing of the children than they ever have been. And so I think that the child issue, of course, puts more pressure on women than men simply because they have to bear them and then they have to feed them. And I see that, but I think generally speaking, making it easier for young people to have children, irrespective whether they're men or women, is a good thing, right? It helps. Do we do a lot? I would actually say in science, yes, we do. I think having daycare, having actually quite a liberal work schedule is really quite important. If you talk to people who work in high-powered firms, law firms in New York, they can't go home at 3 and spend a few hours with their child, put him or her to bed and come back to work. It's very difficult. So I think at some level, as scientists, as academics, we're really blessed by having that liberty. And so I think making it better, but making it better for all parents, I think will only do good, irrespective of, not especially in a sex specific.
So you're optimistic. Your prognosis is that we will get better.
It always does.
Okay, perfect. Let's
Thank
end
you.
there.
Okay, we have